Estimation of hemoglobin by Sahli. This is the Sahli's hemoglobinometer kit, which contains Sahli's tube, a comparator box, and a Sahli's pipette. A closer look at the Sahli's hemoglobin tube. It has got two kinds of readings. First is by gram per deciliter, which is most frequently used, and another is by percentage method, where 100% means 14.5 grams but is rarely used. This is the comparator box with color strips on it, which are brownish in color, and on the back you have a white acrylic plate. We place the tube into the comparator box like this, with the plain side of the tube facing us. We also have a transparent stirrer to stir. We also have a hemoglobin pipette, which has got a marking of 20 microliters or 20 cubic mm. On the pipette you have your tip, you have a 20 microliter marking, and at the back you have attached a rubber tube with a mouthpiece. The mouthpiece goes into the mouth and can be used to suck or blow any liquid. Apart from this, we also have other liquids which are not included in the kit. One is distilled water and another is N/10 hydrochloric acid. Each of them have specific use, which we will see in the procedure. Moving on to the procedure, we take the Sahli's tube and fill it with N/10 HCl up to the lowest yellow. And then we move on to place the tube back into the comparator box. Now we need to prick the finger and get a big generous drop of blood, like the one lying on the slide right now. We take this drop of blood and dip our pipette into this drop and try to slowly suck in the blood up to the 20 microliter mark. Do not worry if you have sucked extra amount of blood. You can always blow some air and get the extra amount of blood out and get the exact 20 microliter blood by blowing and sucking. Now clean the pipette with a tissue paper to get any extra amount of blood that might be lying on the surface of the. Now we will empty all the blood in the pipette into the tube by blowing the blood out into the tube. We also then dip the pipette into the N/10 HCl and take in a little bit of solution, blow it out, and doing this multiple times so as to wash off any blood that might be remaining into the pipette. After we obtain a complete solution, we tap it across our hands and we shake it a little bit to mix it properly. We let this solution stand into the comparator box for 10 minutes and let them react with each other. The HCl and the hemoglobin in the blood react with each other to give a compound known as acid, which is brown. We will now use the distilled water to dilute the acid hematine solution to match the brown color in the comparator box. To take readings, hold it at the eye level across a light source or a window for a proper lighting. We will now use the distilled water to dilute the acid hematine solution. We will add 1 to 2 drops of distilled water into the tube, stir it with the glass stirrer, then pull the glass stirrer up until it is above the level of the solution. Please make sure that the stirrer is just lifted above the solution, but it is not out of the tube. If we pull the stirrer out of the tube, it carries along some of the solution with it. It causes wastage and gives us a false reading. So throughout the solution, throughout this procedure, we will keep the stirrer inside the tube. We will keep repeating this process by gradually adding 1 to 2 drops of distilled water and comparing them with the standard color bars. When the color of the solution is almost same as that of the color bar, we take the tube out, and we look at the lower meniscus of the solution to obtain a reading in grams per deciliter. Further, we add another 2 drops of water, which should give us the exact color that matches the color bars. That will be our second reading. And further than that, we add another 2 drops of water that will give us a little lighter solution color. That will be our third reading. We will take the average of these 3 readings to arrive at our final level. These are the physiological values of hemoglobin for adults.